In 1991, a team of archaeologists excavating a Bronze Age site in the Levant made a discovery that quietly rewrote the history of human nutrition. Buried alongside pottery, tools, and the remnants of a civilization that had collapsed 3,000 years earlier, they found food. Not fossilized traces. Not chemical residues. Actual food. Seeds, dried fruits, pulses, and pressed oil residues still clinging to the interior of ceramic vessels. When they brought in nutritionists to analyze what these people had been eating, the results were unexpected. By almost every modern metric, the ancient diet they reconstructed was superior to the standard diets of the industrialized nations doing the excavating. Higher in fiber. Higher in polyphenol. Higher in essential minerals. Lower in refined carbohydrates. Richer in the specific fatty acids that modern medicine now links to brain health and cardiovascular protection. We had been looking for the future of nutrition. We found it in the past. Here is what that discovery represents at a deeper level. The foods preserved in scripture and sacred texts across the ancient world were not chosen arbitrarily. They were selected, cultivated, traded, and protected across generations precisely because something observable happened when people ate them. Before clinical trials. Before biochemistry labs. Before peer-reviewed journals. There were thousands of years of human beings paying close attention to what kept them strong, what helped them heal, and what allowed some communities to outlast catastrophic disease and hardship while neighboring populations collapsed. Today, we are going to explore 20 of those foods. Not as religious artifacts or cultural curiosities, but as living subjects of modern scientific investigation. Because in laboratory after laboratory around the world, researchers keep arriving at the same remarkable conclusion. The ancient nutritional instinct was correct. These foods work. And understanding why they work changes how you think about every meal you eat. Stay with me especially for number 14, because archaeologists found it packed into the tomb of a pharaoh, and modern hospital wards in 3 continents now use a refined version of it as a wound treatment. And number 2 has a property so chemically unusual that microbiologists still argue about exactly how it manages to kill bacteria that have defeated every synthetic antibiotic we have thrown at them. And number 1 is going to completely reframe how you think about the most common cooking ingredient in the world. Before we go further, if you are the kind of person who finds the intersection of ancient wisdom and modern science genuinely interesting, subscribe and turn on notifications. Every week we go deeper into the food, the history, and the research that most people never hear about. You do not want to miss what is coming. Let us start. Olive oil has been pressed from fruit since at least 6000 BC. That makes it older than the wheel. Older than writing. Older than most of the civilizations we consider ancient. The olive tree itself was so central to Mediterranean life that when ancient armies wanted to devastate a region completely and permanently, they did not burn the houses. They cut down the olive trees. Because the trees took decades to mature, destroying an olive grove was a generational act of warfare. It said, "We are not just defeating you today. We are taking away your future." That is how important this oil was. Modern nutritional science has spent considerable resources understanding why. The answer lies in a class of compounds called polyphenol, and olive oil contains them in extraordinary concentration, particularly in high-quality cold-pressed varieties. The specific polyphenol that has most captured researchers' attention is called oleocanthal. Its discovery, in 2005 by Manel Chemical Senses Center researcher Paul Breslin, came about by accident. He was studying something else entirely, and noticed that extra virgin olive oil produced a distinctive throat irritation. That sensation, he realized, was pharmacologically identical to the effect of ibuprofen. Not similar. Identical. Oleocanthal inhibits the same inflammatory enzymes that ibuprofen targets, through the same biological mechanism. What that means in practice is that consuming quality olive oil daily provides a low-level, continuous anti-inflammatory effect throughout the body. Not the sharp pharmaceutical spike of a painkiller, but a sustained, gentle reduction in the inflammatory signaling that underlies cardiovascular disease, neurodegeneration, and most chronic conditions that kill people in the modern world. The Mediterranean diet studies, and there are hundreds of them at this point, consistently show that populations who consume olive oil as their primary fat source had dramatically lower rates of heart disease, several cancers, type 2 diabetes, and cognitive decline. One landmark trial called PREDIMED, involving over 7,000 participants, found that adding generous amounts of extra virgin olive oil to an already reasonable diet reduced major cardiovascular events by nearly 30%. The key word in all of this is quality. The oleocanthal and other beneficial polyphenol exist in abundance only in fresh, cold-pressed, unfiltered oil. They are sensitive to heat, light, and time. The oil that ancient peoples used was harvested, pressed, and consumed within a relatively short window. It was genuinely a living food. What many people purchase in supermarkets today, heated during extraction, stored in clear glass or plastic, sitting on shelves for months or years, is a very different chemical product. The ancient world used olive oil on everything. In food, in wounds, in lamps, in religious ritual. Its ubiquity was not cultural accident. It was the practical expression of something observed over thousands of years. Now let us move to something equally ancient and equally misunderstood. Raw honey is perhaps the oldest medicine on earth. There is evidence of human honey collection going back at least 8,000 years, depicted in cave paintings in Spain and India. Egyptian medical papyri, some of the oldest medical documents in existence, list honey as an ingredient in dozens of preparations for wound care, eye infections, digestive complaints, and respiratory conditions. For a long time, the scientific community was politely skeptical of honey's medical reputation. It seemed too simple, too sweet, too much like folk belief dressed up as medicine. Then researchers started examining it properly, and the skepticism evaporated. Raw honey is antimicrobial through at least 3 independent mechanisms working simultaneously. First, its low water content and high sugar concentration create an osmotic environment that pulls moisture out of bacterial cells, essentially dehydrating them. Second, the glucose oxidase enzyme in honey reacts with water to produce hydrogen peroxide continuously, providing a sustained disinfecting effect. Third, and most surprisingly, honey contains a small defensin protein produced by bees called defensin 1 that directly attacks bacterial cell walls. Studies have found that this protein is active against methicillin-resistant Staphylococcus aureus, the antibiotic-resistant hospital infection known as MRSA, that costs tens of thousands of lives every year in the United States alone. Manuka honey, produced by bees that forage on the New Zealand tea tree, has such extraordinary antibacterial potency that it has been approved as a clinical wound dressing in Europe, Australia, and the United States. Hospitals use it on chronic wounds, burns, and surgical sites that have failed to respond to conventional treatment. But the critical distinction is between raw honey and commercially processed honey. To produce the clear, indefinitely shelf-stable product familiar from grocery stores, manufacturers heat honey to temperatures that destroy the enzymes responsible for hydrogen peroxide production, filter out the pollen and wax that carry additional compounds, and in some cases dilute it or adulterate it with corn syrup. What remains tastes similar, but functions very differently at the biochemical level. Here is a detail that captures how remarkable raw honey actually is. Archaeologists have found honey in sealed ceramic vessels in Egyptian tombs dating back 3,000 years. It was still edible. Not just technically not rotten. Edible. No food on Earth has a comparable shelf life, because no food on Earth has that combination of antibacterial mechanisms working continuously to preserve itself. Let us talk now about something that illustrates a recurring pattern in this whole story: the gap between how a food looks and what it actually does. Barley looks unimpressive. It is a small, pale grain. Humble. The food of working people, of soldiers on long campaigns, of sailors provisioning for extended voyages. When nutritional science developed the concept of prestige foods in the 20th century, barley was not on the list. Wheat got the attention. Rice got the attention. Barley was quietly ignored. This turned out to be a significant oversight. Barley contains the highest concentration of a specific type of soluble fiber called beta-glucan of any grain on Earth. Beta-glucan is now one of the most thoroughly documented functional food compounds in nutritional science. The evidence for its cholesterol-lowering effects is so robust that the United States Food and Drug Administration permits food manufacturers to make a specific health claim on product labels if they contain sufficient amounts. That is an extraordinarily high bar. The FDA rarely allows health claims on food. The evidence for beta-glucan met it. Here is how it works. When you consume barley, the beta-glucan dissolves in the gut and forms a thick viscous gel. That gel slows the entire digestive process. It slows glucose absorption, preventing the blood sugar spikes that drive insulin resistance. It traps cholesterol and bile acids and carries them out of the body before they can be reabsorbed. It feeds the beneficial bacteria of the microbiome selectively, promoting the species associated with reduced inflammation and better metabolic function. Studies comparing barley directly with oats, already considered one of the healthiest grains available, found that barley produces a lower glycemic response and greater cholesterol reduction. Oats have excellent marketing. Barley has better data. The populations that ate barley as a staple were not making a compromise. They were, without knowing the mechanism, making one of the best nutritional choices available to them. Now here is something that almost nobody talks about when discussing ancient nutrition, and it changes the entire conversation about wheat. When modern people experience digestive problems with bread and pasta, the reflexive response is to blame gluten. The conversation rarely goes deeper than that. But here is the question that nutritional historians have been asking. If wheat is inherently problematic for human digestion, why did wheat-based diets sustain some of the most physically capable populations in human history for thousands of years without apparent epidemic levels of digestive disease? The answer is that the wheat they were eating was not the same wheat we are eating. Modern commercial wheat was developed in the mid-20th century through selective breeding aimed at maximizing yield per acre. It succeeded spectacularly at that goal. Global wheat production increased enormously, helping to prevent famines. But in the process, breeders created a grain with a dramatically altered chromosome count, a different gluten-protein structure, higher starch content, and a significantly reduced micronutrient profile compared to ancient varieties. Emmer and einkorn, the wheats of the ancient world, have between 14 and 28 chromosomes. Modern commercial wheat has 42. This is not a minor variation. It represents a fundamentally different plant with different proteins, different digestibility, and a different effect on the immune system. Studies comparing immune responses to einkorn versus modern wheat proteins have found meaningful differences in how the gut handles them. Many people who react poorly to modern wheat tolerate einkorn without issue. Einkorn also contains significantly more beta-carotene, lutein, riboflavin, and total antioxidants per serving than modern varieties. Its starch is more slowly digested. And because ancient bread was almost universally made through slow fermentation rather than commercial yeast, the lactic acid bacteria in the sourdough culture spent hours breaking down both the gluten proteins and the phytic acid that would otherwise interfere with mineral absorption. The bread that emerged from that process was nutritionally richer and more digestible than almost anything sold in a modern bakery. The grain of the ancient world was not what is making people sick. What is making people sick is what we turned it into over the last 70 years. Let us shift to something that illustrates the extraordinary precision of ancient nutritional knowledge. When the ancient Israelites were traveling through the wilderness and ran out of the vegetables they had known in Egypt, the emotional response recorded was grief. Not mild disappointment. Grief. And the foods they specifically mourned included garlic and onions. If you understand what garlic and onions actually do, that grief becomes scientifically understandable. Garlic produces its most powerful compound through a chemical reaction that only occurs when the cell walls are broken. When you crush or chop a garlic clove, an enzyme called alliinase comes into contact with a compound called alliin. They react instantly to form allicin, one of the most potent natural antimicrobials ever identified. Allicin is active against bacteria, viruses, fungi, and certain parasites simultaneously. Clinical trials have found that people who consume garlic regularly experience significantly fewer and shorter respiratory infections. Multiple studies show measurable reductions in blood pressure and LDL cholesterol with consistent garlic intake. The ancient world used garlic medicinally with confidence. Egyptian medical records, Hippocrates, Pliny the Elder, Dioscorides. All of them describe garlic for infections, gut complaints, and vascular conditions. They did not know about allicin or alliinase. They knew what happened when people ate garlic, and that knowledge was robust enough to sustain itself across thousands of years and dozens of independent cultures. Onions work through related but distinct chemistry. They are one of the richest dietary sources of a polyphenol called quercetin, which has well-documented anti-inflammatory, antihistamine, and antioxidant effects in clinical research. But onions have a second function that the ancient world was practicing intuitively without understanding the mechanism at all. Onions are extraordinarily rich in inulin-type fructans, a category of prebiotic fiber that specifically nourishes the most beneficial bacteria in the human colon. Bifidobacterium and Lactobacillus species, the same bacteria now sold in expensive probiotic supplements, thrive on the compounds in onions. A diet built around garlic, onions, leeks, and related plants was building and maintaining a healthy gut microbiome by default. Before anyone had a word for the microbiome, before anyone understood that the gut housed the majority of the immune system, the ancient diet was feeding it correctly. The grief over lost onions and garlic was the grief of people who, on some level, understood that they were losing a protection they could not afford to lose. Now I want to talk about something that most people overlook entirely because it seems too exotic. But stay with this because the science is remarkable. Pomegranates appear in ancient texts across the entire arc of the biblical world, from Egypt to Persia, from Greece to Rome. They were carved into temple architecture, woven into priestly garments, used as motifs in royal jewelry. This was not purely aesthetic admiration. In a world where visual symbolism almost always carried functional meaning, the universal reverence for the pomegranate tells you something significant was observed about what this fruit did. The compound that has most captured modern researchers' attention is punicalagin, found in meaningful concentration almost nowhere else in the plant kingdom. In the gut, punicalagin breaks down into compounds called urolithins, particularly urolithin A, which has shown remarkable effects in laboratory and clinical research. Urolithin A appears to trigger a process called mitophagy, the cellular housekeeping mechanism by which the body clears out damaged mitochondria and replaces them with healthier ones. Mitochondrial dysfunction is now understood to be a central feature of aging, neurodegeneration, and metabolic disease. A compound that specifically promotes mitochondrial renewal is of enormous interest to longevity researchers. Clinical trials on pomegranate juice have shown reductions in systolic blood pressure, improvements in arterial elasticity, and decreases in oxidized LDL cholesterol, the specific form of cholesterol most directly involved in plaque formation. Its antioxidant capacity in standardized tests consistently outranks green tea, red wine, and blueberries, which are the foods most commonly associated with antioxidant richness in popular nutrition culture. And the rind, which most people discard without a second thought, contains the highest concentration of antimicrobial tannins in the entire fruit. Traditional healers across the Middle East brewed it specifically for intestinal infections and parasites. Modern microbiology has confirmed those tannins are genuinely active against several gut pathogens. The ancient world was right about the pomegranate. It just did not have the vocabulary to explain why. Let us talk about figs, because there is a story here that most people know without realizing what it actually means. When the ancient Israelite king Hezekiah was dying from what ancient texts describe as a severe infected ulcer, the healer who attended him did not rely solely on prayer. He prescribed a poultice of pressed figs applied directly to the wound. The king recovered. Figs contain a proteolytic enzyme called phycin that breaks down dead and damaged protein tissue. Proteolytic enzymes are the active mechanism in modern medical wound debridement, the clinical process of removing necrotic tissue from infected wounds to allow healing to proceed. The fig poultice was performing a version of a procedure that modern medicine still relies on, using chemistry the plant had developed for its own purposes over millions of years of evolution. Beyond wound care, figs are extraordinarily dense in soluble fiber, specifically pectin, which is one of the most effective prebiotics available in whole food form. They deliver calcium, magnesium, potassium, and B vitamins in concentrations that make them one of the most mineral-rich fruits studied. In a world without vitamin supplements, a reliable supply of figs was a genuine nutritional asset. The fig also carries an interesting footnote in the history of human self-knowledge. It is one of the first plants mentioned by name in Genesis. Ancient peoples who lived close to food understood its significance in ways that we, insulated from agriculture by supply chains and supermarkets, have largely lost. Now let us turn to a plant that illustrates something fascinating about the relationship between religion, ritual, and practical medicine. Hyssop appears in the ancient texts repeatedly, always in the context of purification. Sprinkled on people emerging from illness. Used to cleanse spaces where disease had been present. Applied in ceremonies meant to restore someone to a state of cleanness after contamination. For centuries, biblical scholars treated this as purely symbolic. The idea of ritual purity as a spiritual concept without physical correlate. Then biochemists started analyzing hyssop and found something that reframed the entire discussion. Hyssop contains carvacrol and thymol in significant concentration. These are the same primary antimicrobial compounds found in oregano and thyme, and they are among the most potent naturally occurring antiseptics known to science. Studies have confirmed that hyssop extracts inhibit a broad range of bacteria and several viruses including herpes simplex. Its expectorant compounds actively loosen and clear mucus from the respiratory tract. Its anti-inflammatory properties explain its traditional use for lung infections and inflammatory skin conditions. When you understand this, the purification rituals acquire an entirely different dimension. Sprinkling hyssop on a person emerging from disease was not merely symbolic. It was the application of a genuine antimicrobial agent. Using it to cleanse spaces that had housed sickness was a functional sanitation practice. In a pre-microscope world where germ theory did not exist as a concept, someone had worked out empirically that this specific plant, applied in this specific way, reduced the spread of disease. And they encoded that knowledge into religious practice to ensure it would be preserved and followed. That is not primitive thinking. That is sophisticated epidemiology in the only language available to express it. Cumin operates similarly, though its story is nutritional rather than antimicrobial. The fact that it was valuable enough to be tithed in the ancient world, set aside as a religious obligation alongside more obviously precious goods, tells you how the people who used it daily understood its worth. Modern research has confirmed cumin's effects on blood sugar regulation, with multiple studies showing it improves insulin sensitivity and reduces fasting glucose in people with type 2 diabetes. Its compound cuminaldehyde has demonstrated antimicrobial activity across a wide range of pathogens in laboratory conditions. It stimulates bile production and digestive enzyme activity, which improves the absorption of nutrients from every other food eaten alongside it. That last property is particularly interesting. Cumin was used in cooking not just for flavor but because, at some intuitive level rooted in observed experience, it made the food work better in the body. 3 grams of cumin, a small amount, provides approximately 20% of daily iron requirements. In populations where iron deficiency was a genuine threat to energy and immune function, a spice that was present in nearly every meal was providing meaningful nutritional insurance. Coriander seed, the dried form of what we call cilantro, tells a similar story. It was found in sealed containers in Tutankhamun's tomb, indicating it was valued enough to accompany a pharaoh into the afterlife. It appears in ancient Egyptian medical texts, in Sumerian records, in the earliest Sanskrit writings. No single culture invented coriander seed as medicine. It was independently recognized across multiple civilizations as something that reliably helped digestion, eased inflammatory pain, and calmed anxious nerves. That last observation has attracted serious scientific attention. Animal studies comparing coriander seed extract to pharmaceutical diazepam, a prescription anti-anxiety medication, found comparable anxiolytic effects at certain doses. The mechanism appears to involve the same GABA receptors in the brain that benzodiazepine drugs target. Ancient physicians who observed that coriander calmed agitated patients were recording a real pharmacological event without the vocabulary to describe its mechanism. The emerging research on coriander and heavy metal detoxification is still preliminary, but the signal is intriguing. Several studies suggest that cilantro, the same plant in its fresh form, may help mobilize mercury and other heavy metals from tissues. Given that ancient urban populations lived with significant metal exposure from water storage vessels, cooking implements, and industrial processes like smelting, a plant that helped clear those toxins from the body would have had observable effects on health that would reinforce its medical reputation. Let us spend a moment on fish, because the nutritional decision to center a diet around small fatty fish from a freshwater lake, which is what the diet of first-century coastal communities in the region appears to have been, turns out to be one of the most beneficial choices a population could make. The omega-3 fatty acids found in fatty fish, specifically EPA and eicosapentaenoic acid and DHA or docosahexaenoic acid, are not optional additions to human nutrition. They are structural requirements. DHA forms a significant component of the membrane of every neuron in the brain. EPA has powerful anti-inflammatory effects throughout the cardiovascular and immune systems. The human body cannot synthesize either in meaningful amounts. They must come from diet. The research on omega-3 sufficiency is some of the most consistent in nutritional science. Populations with high fish consumption show dramatically lower rates of cardiovascular disease, depression, postpartum mental health problems, cognitive decline with aging, and inflammatory conditions. The mechanism is clear. The evidence is large and consistent across diverse populations. The fish of the ancient Galilean diet were eaten whole, simply prepared, unprocessed. No batter. No industrial seed oil frying that chemically alters or destroys omega-3 content before it reaches the body. The nutrition was preserved because the preparation respected the food's chemistry, even without understanding it explicitly. Now let us talk about a nut that a biblical patriarch considered valuable enough to send as a diplomatic gift to the most powerful ruler in the ancient world. When Jacob needed to make a favorable impression on the Egyptian official controlling grain distribution, during a regional famine, he sent a selection of the finest things his land produced. Among them, pistachios. That detail is not incidental. In the context of a desperate famine, you do not include a food in a diplomatic gift unless that food carries genuine prestige. Pistachios did. Modern nutritional research explains the prestige. Pistachios are uniquely high in lutein and zeaxanthin, carotenoids now known to be essential for long-term eye health and specifically associated with protection against macular degeneration, the leading cause of age-related vision loss. In a world where functional vision was directly connected to the ability to work, hunt, navigate, and survive, foods that preserved eyesight would have been observed, valued, and coveted. Pistachios also stand out among nuts for their prebiotic fiber content. Research has found that regular pistachio consumption significantly increases the population of beneficial bifidobacterium in the gut, more so than most other commonly studied nuts. They contain meaningful amounts of resveratrol and other polyphenol associated with cardiovascular protection. Clinical studies have shown improvements in blood sugar control and lipid profiles with regular pistachio consumption. The patriarch who sent them as a gift to a pharaoh was demonstrating an intuitive understanding of their value that modern biochemistry is still cataloging. Almonds carry their own remarkable story. They are extraordinarily rich in vitamin E in its most bioavailable form, alpha-tocopherol, a fat-soluble antioxidant that protects cell membranes throughout the body. They deliver magnesium at a level meaningful for a population where magnesium deficiency, even today, affects a large proportion of adults eating modern diets. They provide monounsaturated fats, protein, calcium, and fiber in a naturally balanced ratio. What is genuinely interesting about almonds is a property discovered through careful clinical measurement. The actual caloric absorption from almonds is lower than the label suggests, because the cell wall structure of the almond traps a portion of its fat content and passes it through the gut intact, where it feeds beneficial bacteria rather than being absorbed. The ancient practice of eating raw almonds as a staple food was providing more prebiotic benefit and fewer net calories than anyone calculating on paper would have predicted. Let us come now to the plant whose story I have been saving, because it ties together several themes and ends with something genuinely surprising. Carob has been eaten across the Mediterranean and Middle Eastern landscape for at least 4,000 years. The pods of the carob tree are sweet, chewy, naturally shelf-stable, and nutritionally extraordinary. In the ancient world, they were known by several names depending on the culture and language. One name, used across the Levant and adopted into multiple traditions, translates roughly as locust bean. This matters because it changes the reading of one of the most famous dietary descriptions in ancient literature. John the Baptist, living in the Judean wilderness, subsisting on what the text describes as locusts and wild honey. Biblical scholars have long noted that the carob tree, growing wild throughout exactly the region where John ministered, was routinely called locust bean by the local population. What reads to modern eyes as an insect-based diet may, in the original cultural and linguistic context, describe two of the most nutritionally powerful foods the ancient wilderness provided. Carob pods contain roughly 50% natural sugars packaged with up to 40% dietary fiber by dry weight. That combination, fast energy paired with exceptional fiber, produces exactly the sustained, slow-burning caloric release that would allow someone to walk long distances, speak loudly to large crowds, and maintain the physical energy the life of an itinerant teacher in difficult terrain would require. The fiber in carob is primarily insoluble, making it a powerful regulator of gut transit. It was used in European hospitals within living memory for childhood diarrhea caused by rotavirus, with clinical studies confirming its effectiveness against gut pathogens. The condensed tannins that give carob its mild astringency have documented antimicrobial properties that help explain both its traditional medical uses and its extraordinary shelf stability. combined with raw honey, which provides its own antimicrobial protection and fast-acting sugars. Carob and honey represent a survival diet of remarkable comprehensiveness, not a deprivation diet. A diet that, judged on nutritional and medical criteria, outperforms most of what the wealthiest people in the ancient world were eating in their palaces. Sometimes the wilderness diet is the wise diet. Let us now step back and look at what all of these foods share, because the pattern is not accidental. Every food on this list is whole. Every food is minimally processed. Every food contains its original complement of fiber, polyphenol, minerals, and enzymes intact, the way the body evolved over hundreds of thousands of years to receive them. Every food was grown in soil that had not been depleted by industrial monoculture, was not treated with synthetic pesticides that alter the plant's own chemical defenses, and was consumed relatively close to the time and place it was produced. The ancient world did not eat this way because they were enlightened about nutrition science. They ate this way because it was the only way available. The processing, the refining, the extraction, the stabilization that transformed these foods into the pale nutritional shadows sold in most modern grocery stores happened because technology made it possible and economics made it profitable, not because it was better for the people eating it. What the research keeps telling us, in study after study, in trial after trial, is that the further we move the food from its original form, the more we lose. The olive oil that is cold-pressed and consumed fresh is different from the oil that has been heated, deodorized, and stored for a year. The wheat that has been fermented slowly for 12 hours before baking is different from the bread produced from fast-acting commercial yeast in 90 minutes. The honey harvested from a local hive and eaten raw is different from the processed product that shares its name, but not its chemistry. The ancient writers who recorded these foods in their texts were not writing nutrition guidelines. They were writing about life, about what sustained people, what healed them, what they mourned when it was taken away, and what they celebrated when it was abundantly present. The nutritional and medical significance was embedded in the culture, the religion, the economy, and the poetry simultaneously, because these things were not separate domains. They were all expressions of the same observed reality. Modern science is not discovering something new when it validates these foods. It is finally developing the vocabulary to explain something that human experience had already documented across thousands of years and dozens of civilizations. The question the research raises is not whether these foods worked. That question has been answered. The question is why we stopped treating them as the extraordinary things they are. You do not need to overhaul everything at once. Start with one food from this list. Understand it. Find the best quality version you can. Add it to your routine deliberately. Then add another. The ancient table was not built in a day. It was assembled across generations of careful observation and accumulated wisdom, and it is available to you right now, more thoroughly documented and more scientifically validated than at any point in human history. The people who wrote these ancient texts paid attention to what healed them and what sustained them across lifetimes. That attention was the original research. We are still catching up. If something in this video shifted the way you think about the food on your plate, leave a comment below. Tell me which food surprised you most, or which one you are going to start with. I read every comment. If you want to go deeper into this territory, the history, the science, and the practical application, subscribe and hit the notification bell. We go further every week, and the next video is going to take this conversation somewhere unexpected. See you there.